Okay, so we'll be starting in a couple of minutes. And few of the participants have introduced yourself in the chat box. And I'm also curious to know, do you have any specific research interest within bioinformatics? Aishi says she wants to do metagenomics approach in bioinformatics. I'm monitoring the chat. Let me know what are your research interests with respect to bioinformatics. Are you still looking for a particular area of research, or do you have a particular idea in your mind? Let me know in the chat. We'll be starting in 5 more minutes. In the meantime, all right, so let's start with today's session. So hello everyone, a very warm greeting to one and all. My name is Sri Gauri Krishnakumar and I would like to welcome you all for the very first session on Introduction to Bioinformatics Multi-Omics. This training program is designed for everyone, including students who don't have a background in bioinformatics, as well as life science researchers. And the objective is to introduce topics and examples to help participants understand omics data and the use of bioinformatics in life science research. In today's session, we will gain a brief overview about the educational materials and projects, as well as the T-Bioinfo platform that you will get access to as a part of the program. And before we begin with today's webinar, for those of you who are joining for the first time, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of the program, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. This training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data. Students will also have access to the Omics Logic resources, which takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And no matter what the user's initial experience with bioinformatics is, Omics Logic is going to be a very useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, an introduction to basic coding languages like R and Python, and even beginning a data analysis project. And this also comes with an access to the Omics Logic courses, which includes courses on bioinformatics, precision medicine, multi-omics that consist of genomics, transcriptomics, metagenomics, and epigenomics, as well as data science that makes use of bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. And another important resource that you get access to is the T-Bioinfo platform. Users will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. And this platform includes demo pipelines, as well as data management and analysis cloud infrastructure to run the bioinformatic pipelines. And different stages of analysis are performed in different sections on this Multi-Omics platform. So now to view the various courses and example projects and student projects, you need to first sign up on the Omics Logic Learn portal. For that, let me walk you through on a brief demo on how you can sign up. So I'm pasting the link in the chat box for the participants to sign up on the Omics Logic Learn platform, where you will be able to gain all the access to the various courses that I was showing to as part of the program. So for those participants who are signing up on the Omics Logic Learn portal for the very first time, you can sign up either using either one way, that is enter your email ID and provide a password. You can click on the create an account option that you see here and enter your name and email ID and a password. This is one way how you can sign up. Another way you can sign up is using any of your social media handles. This could be your Google, Facebook, Apple account, GitHub account, or even Twitter. And for those participants who have already enrolled for the program and who already have an account on the portal, you can simply click on the login now option that you see here and simply enter your email ID and password that you used to register on the portal and click on sign. So I'll pause for a couple of seconds for the participants to sign up, and I'll walk you through the demo on what are the various courses and example projects that are there. So please let me know in the chat. I'm monitoring the chat box for your responses. Once you have signed up on the portal, please put a yes. And if you already have an account on the portal, please put a done in the chat box. Aishi has signed up. Pankaj has already signed up. Charles has also signed up. What about the rest of you guys? Okay, we have a lot of responses. Shay Rosen. If you are facing any queries, you can let us know in the chat as well. So I can see that a lot of the participants are signing up. So please continue to put your responses in the chat box. Due to time constraints, I'm just going to sign in using my login credentials. So once you sign up on the portal, you can see this interface where you'll be able to see the various courses and example projects that are there. So if you scroll down below, you will see a search bar that says looking for a specific course. So in this tab, you can search for a specific area of interest that you have. So let's say you are interested in oncology. So you can simply enter oncology and that will show you the various courses and example projects that are related to cancer. And I can see that a lot of the participants might be interested in introductory bioinformatics. So you can simply enter the term bioinformatics, and that will also show you the various introductory courses that are there in bioinformatics. So you can browse through at your own pace and have a look at the various courses and example projects that are there on the portal. So for now, I'm going to show you how you can set up your profile on the portal. For that, you can simply click on the tab that says welcome back with your email ID. So once you click on this, you will be able to see your account settings. So here, you will see an icon that says profile. Click on that, and that will show you how you can edit your profile. So as you can see here, the very first important feature that you need to update is your profile image as well as your name. Make sure your name has to be in proper capitalization and not that all your name is going to be in capital letters. And also you have the option to link your social media handles. So in my case, I have linked my LinkedIn account and my org ID. And another important feature that you must update on your profile is your bio. So here is an example bio that I have. That is, I have mentioned my educational background and my research interest. So you can also do the similar for your bio. And you can also see various tab options in your profile. Under the activity tab, the program mentor and the community manager will be tracking your learning progress. And under the courses tab, the courses that you are currently completing will be displayed. So for in my instance, in my case, I'm currently completing transcriptomic analysis in Python. And finally, once you complete a project and publish it on our portal, that will be displayed under the projects tab. And this is the programs tab where you will be able to get access to your program information. So in my case, you can see I have enrolled for 2 programs. One is precision medicine and one is for the research fellowship. Finally, after the completion of your courses, the required assignments and quizzes, you will be able to receive your certificate for the course under the certificates tab. And another interesting tab that I want to show you is the projects tab. So once you click on this, you will be able to see the various student projects that are there that have been published on our portal. So the projects topics range from oncology, infectious diseases, precision medicine, machine learning, and so on. So you can have a look at all of these at your own pace. So that was all about setting up your profile and navigating through the courses and example projects that are there. So now we can start with the today's session. For that, I'd like to pass on the stage to Shubham, who will be playing a short video recording by the program. So over to you, Shubham.
Thank you, Sri Gauri. I'm just sharing my screen.
Perfect.
Could you confirm is my screen is visible?
Yes, we can see your screen.
All right, so I'm just starting the session.
Sure. Hello, everybody. So welcome to today's first session on Introduction to Bioinformatics program. So this program is an introduction to the field of bioinformatics, or in this program, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that come with widely accessible, high-throughput technologies and computational methods for analysis, for annotation of such detailed data in biomedical research, in pharma industries, and elsewhere. We will also discuss various data types. We will discuss emerging technologies and their impact on healthcare, environmental sciences, public health, agriculture, and etc. etc. So let's learn more. About the program and the curriculum of this program, and what do different sessions cover, and how are they structured, and what are the resources that you are going to be accessing in order to understand or in order to use this program when you travel through this program. So this program has 10 sessions, and in these sessions you will see a broad applications of bioinformatics, next generation sequencing, biostatistics as a tool for big data analysis. So we also have lots of practical assignments in which we will be learning about the logic behind this bioinformatics analysis, and to see how these data science methods can be used to study or used to be applied to biomedical research in general. So when you complete this program, you will also be able to plan how to take what you have learned and how to apply it to an independent research projects using the logics you understood, using the tools we introduced you, and using the methods we cover in this program. So first, let us start with an obvious question on the place of bioinformatics approach in biomedical research. So one of the primary driving forces for the necessity of bioinformatics is availability of the data that is generated in huge volume. So the volume of data kind of changes the biological domain to a data-driven science compared to the traditional hypothesis-driven nature of the biomedical research. The data and its availability leads to the fact that we have some method of structuring, or we have to devise some method of structuring and organizing this data to make sense of the patterns that we are discovering. Bioinformatics approaches can also in fact refer to methods of analysis, and that will help us in the interpretation of the biological data, in the annotation of the biological data with respect to the phenotypical conditions that we are studying our analysis with, studying our samples. So what is bioinformatics and how do we use it? So it can be summarized or it can be defined as a science of both storing lots and lots of complex biological data and analyzing this data to find new insights, which we will use in many different ways. So bioinformatics is effectively is fundamental to many biological research, and it involves either biologists who learn information technology or mathematicians and statisticians who learn the foundations of biology. So in short, it is a convergence of biology and data where both are equally important, as we will see in the next few slides, and they will be detailed in this program. So today's biomedical research, for example, isn't simply about finding or isn't simply about publishing one set of results and hoping other researchers read it and find connections to all the other links. So it's about finding patterns, it's about discovering patterns in all forms of data and connecting everything that is out there in the biological domain to provide new insights that we can only spot if we see or if we look at the bigger picture. So bioinformatics lets us bring together the data from lots of different experiments into one place. So we can ask those big questions and find the relations and find the answers and communicate to our peers. So such data availability from different sources, or rather, from these sources, has changed the nature of biological research itself, and we observe that the biology itself is rapidly acquiring the characteristics of data science. So when this happens, or when this biology, biomedical research and biological research rapidly acquires the characteristics of data science, we see this is due to the billions and billions of data points on genes and on proteins and other molecules that are compiled in large files and are systematically studied. So analyzing this data will lead to more knowledge and understanding about living organisms, including our health, biotechnology, including about resources for food like crops and livestock, and etc. etc. So this course is designed to give you an overview of bioinformatics for many of your participants, specifically students, biologists, clinicians, faculties, etc. So this influx of data associated, and the associated methodologies and the data-driven research makes bioinformatics for life sciences as prominent as the emergence of computer for tech industries in these days. So this whole field is based on high-throughput technologies that generate billions and billions of data points on genes, on proteins, and other molecules which characterize the biological systems and phenomena. These data can be phenomics, genomics, epigenetics, transcriptomics, and more about this we will discuss in detail in the coming slides and also in this several different sessions in the program. So these data are actually compiled into repositories and can be systematically studied and understood and interrogated to establish trends for many different applications. So if such data and such data with so much volume is available? Yes, it is available, and one such repository is maintained by the National Center for Biotechnological Information, NCBI. And for example, this NCBI database has a collection of peer-reviewed articles, biological data sets, and projects on various topics. As part of biomedical research, we would not have, as a part of biomedical research community, we would not have done anything without referring to the PubMed database that I was defining. So, as you can see, in the past several decades, this resource has been growing exponentially. And to give you a scale, there are over something like 80,000 publicly available data sets in just one part of NCBI called Gene Expression Omnibus, which predominantly stores transcriptomics data for several different sites, as submitted by the users right here. So such repositories containing data sets of thousands and thousands of genes in each sample, with tens or even hundreds of samples per data set for different conditions, are rich sources from which we can leverage to understand the biology of the system, to ask impactful questions, to relate this data with several other physiology and several other biology that is very well established, or that is that to be understood and related. So this program is designed to highlight the role of big omics data that we discussed right now, which is shaping the biomedical innovation, where molecular data is used for precision diagnostics, big data integration, and machine learning are actually transforming the treatment selection or the personalization of the treatment selection, and the presence or application of innovative algorithms allow for unprecedented capabilities with the drug discovery, with the design of large molecules and drug repurposing, and in each and every aspects of biomedical research has been elevated due to the usage of this data and big data and bioinformatics associated with this bit. So this program will cover some of the important technologies and the applications in today's program. So you participants will be able to access bioinformatics resources along with our mentor support to get help with the learning processes, and which will transform or help you learn the transformation of biology into data-driven science. So by the end of this, you will learn about the importance of bioinformatics concepts and applications, various applications of next-generation sequencing, the role of data science for biologies, and industrial challenges challenges that are addressed by big data informatics. So to get started, I would suggest all of you to go through this collection of several basic courses that will primarily introduce you to bioinformatics applications in healthcare, biomedical research, biotechnology, and agriculture. This will also provide you with a concise overview of various concepts that you need to understand in big omics biological data analysis. So in addition to that, those of you who are interested in venturing into coding practices for bioinformatics analysis, this introductory courses will also introduce you to some basic functions in popular coding languages such as R and Python. So if you already know the basics of bioinformatics, if you know what the general idea is, and you would like to get a detailed entry into some special, excuse me, my animation is running out of time. Yeah. So if you would like to get more detailed entry into these specialization tracks, we have multiple courses and programs addressing your needs. So in these specializations, you will get resources to learn more advanced methods of analysis, and you will learn how to extract meaningful insights from larger data sets using code-free option that will employ or that will actually leverage the big data bioinformatics server, the Talbot Bioinfo server, which we will get a lot of introduction and hands-on demo to, or you can also choose to understand the analysis using application in a coding environment with Python or R. So if you want to get more information on these specialization courses, then you should complete these programs first, and then, for example, if you need more information about these specialization courses, then I would suggest you to complete this Introduction to Bioinformatics program first. Then you will be able to decide, or then you will be able to focus on one or couple of specialization after that. So after these programs in which we discuss about the basic data types, skills necessary to analyze these data types, then we have these specialization options as I discussed. And these specialization courses are designed to introduce you to projects sourced from publications, where we take examples from publications or publicly available data sets, and we start discussing about the methods in the context of particular research question. And these are compiled into example projects under different categories like oncology, infectious diseases, neuroscience, and agriculture. So these projects allow you to not only apply the bioinformatics that you learned technically, but also to use it in a way to conduct research, asking questions, to develop hypotheses, and to translate your understanding of the data to a step-by-step process that will give you results that you are looking to address in the area of oncology, etc., in infectious diseases and other cases. Because why is this context important? Because ultimately the methods are actually best learned if we put that into practice in the context of real case studies. So this is where these example projects that are prepared by taking publicly available data sets and to explore them for the application into hypotheses and methods that we will use these data sets to understand both the research challenge and also the kind of analysis that we are actually designing, or we are deciding to extract meaningful information from these complex omics data. So to start with the syllabus, or to start with the curriculum of this specific program, this is divided into subtopics. So first topic that you will get introduced to is genomics, and introduction to genomics. So this program will actually, or these sessions will actually help you understand why we should study genomics. So to give you a context, this is an essential subfield of bioinformatics, and this is one of the major force in understanding or in expanding our knowledge of associating genetic variants with diseases and other abnormalities. So this is developing, this is a field in which development of methodologies for DNA sequencing, as well as analysis methods to analyze these DNA sequence data and of genomics data from next generation sequencing technology. And for example, to relate that to relate this analysis to several different DNA variations and mutations that can be linked to phenotypical variations of the sample, to link to the disease biology, and also to generally link to abnormalities that we can observe from the context of the sample that we are taking. So this understanding of genetic association provided by this genomic research have great value in medicine, ecology, biotechnology, many pharma industries, and also in agriculture. So what are we actually studying, and how are we relating that to these phenotypical associations? So we start with absolute basic, like we start with what is a DNA code, and what is the basic, or what is the fundamental data that we are recording, and the variation that we need to understand. So the variation that we need to understand is the DNA code, changes in the DNA code when we compare it with the reference sequence or the genomic sequence reference sequence of any typical or healthy human being. Go forward, and the effect of these sequence mutations are or will be covered in detail in the program sessions. To give you an overview, for example, any variation in the sequence set or in the sequence that you have sequenced for your sample compared to the reference sequence cannot be pathogenic. We may have variation that will alter the look and the functionalities of specific samples without, for example, leading to harmful results. And these we call them as variations, or generally we call them as variations, or sometimes neutral mutations. But mutation is a specific term that we reserve for associating themselves with a pathogenicity or with the harmful nature or harmful results of the variations or variants that we find in the sequence. Right? For example, in this simple graphic, only the last functional form has a pathogenic mutation because that interferes with the fundamental functionality of this specific life form. So anything that is harmful to the cells or to the samples are termed as mutation, and sometimes when these variations that have insignificance or that does not have significant harmful levels are generally called as variants. This is an accepted terminology in this field. So how to do this specific analysis? We will again use the Bioinfo server to set up the processing and to set up the analysis or to set up mapping of these small reads into the reference genome and to identify the changes in our sample sequence compared to the reference genome and to annotate these variations based on available knowledge in the publications or in the literature or available knowledge in the repositories, common repositories, and we can actually identify those changes or variants that can be related to phenotypical conditions of the sample or abnormalities that we are studying or the disease biology in general. Right? And this data can also be subjected to several other post-processing methods with which we can again extract meaningful information and insights about the biology of the sample. So some of the known or some of the output that you will gain access to or you will generate while performing these analysis methods or genomic analysis pipelines are characterization of variants, one such as this, where we can visualize the mapping of the reads of your samples onto the reference sequence that is given in this gray bar. Since it is zoomed out to a bird's eye view, we cannot read this reference sequence. And we can also understand the strength of alignment of your reads to this particular or specific position of the genome. And these vertical bars in purple or magenta color are the variants that we can or we will characterize or that we will annotate depending upon their severity as quoted in the famous repositories. So to understand more about this genomics data analysis, and during the course of this program also, we will refer to a lot of lessons from this genomics course, and interested participants can check this genomics course for further information. So our next set of sessions will involve understanding or will involve covering metagenomics and microbial abundance analysis. So this course is going to introduce you to bioinformatics analysis, or these sessions rather will introduce you to bioinformatics analysis of metagenomic sequence data. We will start speaking about some of the sequencing technologies and also some of the methods to study microbes and, for example, several different analysis tools for metagenomic studies, and we will also see few examples where these analysis approaches have been used. So to give you a context, microbes inhabit just about every part of our human body. Like it's living on the skin, it's actually living in the gut, it's living up in the nose, living everywhere. So these microbiome, or the diversity of such microorganisms that live or that inhabit human bodies, are estimated to make up more than half of our cells, right? And their diversity is extremely critical to human health or our health. Most of the time, these microorganisms live in harmony with their human hosts, providing vital functions that are essential for the human survival. For example, they can also enable the work of immune system. They can, for example, be linked to their imbalance or their lack of diversity can be linked to nearly every health condition, as we are discovering regularly. So today we seek, for example, in today's research, we seek interventions to manipulate the population of this microbiome from several different sources. One of the popular sources is the gut microbiome, with diverse products such as probiotics, drugs or therapies, changes in the food habits and wellness products, etc. All of this to and manipulate the microbial population so that we can lead a healthy life. So the imbalance is very necessary to address, and this is called dysbiosis of the microbial population that we will get introduced to. So this dysbiosis or imbalance in the microbial diversity, as we know, is related to many modulations of disease biology, infectious diseases, and their pathogenesis, and sometimes to autoimmune diseases and a lot of other health conditions like chronic diseases and etc. To general health, general well-being also, like the ability to digest fat-filled food and risk factors to develop diabetes or chronic diseases and etc. So, the role of these gut microbiome is very actively linked to several health conditions also. So these associations, these promises, and these microbial studies fuels the exponential increase in this availability of this data. Right? There is, for example, significant advancements in and major initiatives in acquiring this microbial metagenomics data from several different parts of the human body. So these studies have, for example, made data sets that are possible to get from the data sets that are extracted from gut, which is popular resource, and skin, lung, and even sometimes brain. So these data sets, and over the past decade, United States has actually spent more and more on microbiome research, not only in the US, but also in the European Union, in China, Canada, Ireland, Korea, Japan, and etc., wherever we have investment that has confirmed the importance of microbiome for human health and development. For example, to give you a context, 20% of the human investment, 20% of the US investment in such research has gone into two major phases of the human microbiome project. Right? And this human microbiome project is creating the resources, the data that is necessary to understand the diversity and their pattern involving human well-being generally. Right? And it is because of this huge investment of capital, and it is because of this huge availability of the microbiome and metagenomics data. Today, we know that, for example, we know that the endogenous human microbial communities contribute to several critical metabolic, to several important physiological and immunological functions, right? They include maturation of the immune system, they include food digestion, energy generation, and nutrition extraction in general. They include metabolic regulation and control of the fat storage. They also include detoxification of the harmful chemicals, and they definitely include maintenance of the skin, and they also are associated with the mucosal barrier function, and they're also, for example, their involvement in in the severity of SARS infection is also studied of late in the context of the pandemic. So in this sessions or in these sessions, we will actually introduce you to several tools that will help you to perform population analysis. Okay, again it is going very fast. Okay, so to population analysis, and these tools will help you to identify microbes without, for example, prior knowledge of what the sample contains, right? And these technologies and these analysis methods, which will enable us to perform sequencing-based examination of this microbial community, has actually indeed become useful and a powerful tool for generating new and novel hypotheses relating microbial population to several of the disease conditions, as we have been discussing, and as these sessions will also introduce you.
Hello everyone. I would like to pass on to Sonalika. Sonalika, could you please?
Thank you so much, Shubham. I think we had an audio trouble. We did not have the audio well played. So we'll email the participants the recording of the session so that they can revisit the topics that were supposed to be discussed, although we have taken up most of the topics. Now I would like to share my screen and discuss about the program. Let us see what all sessions do we have and how are we going to conduct this program in a hybrid way where we will have live sessions conducted, live Q&A sessions with recorded mentor sessions that would be shared for training, and also how we will be taking care of the resources access, and also the portal coursework and the Bioinfo server access that the users would get. So right now I'm on the Bioinformatics program page. I'll just put the link in the chat box. Yes, so on this program page, you can find the complete session schedule of the program. If you scroll down, you can see each one of these sessions, the days for which these sessions are scheduled. Today we had the first session, the topics that would be covered, and the associated online resources that includes lessons and courseworks on the Omics Logic Learn portal that you have to go through as a part of each and every session. And if in case the participants are not able to attend the live session or the live Q&A session because of their prior commitments, then they can go through the recording of the session that would be uploaded under each session as a part of the specific session that you have to take up. Now, these resources are all hyperlinked. So if you click on the resource, you will be redirected to the specific lesson of the course that you are required to complete as a part of this specific session. So this is the first course which introduces you to the field bioinformatics. What is bioinformatics? What is the role of big data in bioinformatics? What is the role of bioinformatics in healthcare? And what is translational bioinformatics? Today, we wanted to introduce people from different fields, biochemistry, microbiology, biotechnology, to the field of bioinformatics, and also introduce you all to big data so that you can begin with the study of omics. In the next session, we will discuss about genomic data studies. We will introduce you all to genomics, DNA variants and mutations, and following which we will take up the session on metagenomics. After metagenomics, we would be focusing on microbiome functional bacterial community and then discuss about transcriptomics. Our motive behind conducting this program is to discuss students, researchers to the field of omics, which includes genomics, transcriptomics, epigenomics, metagenomics, and also discuss about how machine learning and data science tools and techniques could be applied to analyze and visualize different types of omics data to extract biological insights out of it. After the transcriptomic studies, we will take up the session on single cell transcriptomics, which is one of the most evident fields that the researchers are now working on. And then we will discuss about different tools and techniques that could be used in the field of data science and machine learning different types of machine learning, unsupervised and supervised analysis. And finally, we'll also discuss some case studies to understand how the different concepts that we learned could be applied to answer a research question or frame a research question and work on it. So that would be the flow of the different sessions in the program. And we have the dates mentioned, the topics and the resources that the participants are supposed to go through. So this was about the program schedule. And now I would like to pass on the stage to Ms. Sparsh Thar to talk about the registrations. Over to you, Sparsh.
Thank you so much, Sonalika. I'll just share my screen and I'll quickly navigate you all through the steps of registration. So Sonalika has, okay, just a moment. Sonalika, could you please confirm if you can see my screen?
Yes, Sparsh, your screen is visible.
Perfect. So Sonalika has already shared the link to the program page with you in the chat box. And once you'll open up the link, you will see like the program has already commenced today. Today was the first session. And in this first session, the agenda was to tell you about the different resources which we have, the demonstration of the like communication channels, either Slack, Telegram, or maybe WhatsApp groups, etc. And then tell you about the different platforms which you'll have access to and what all topics and curriculum which we'll be following throughout the program, throughout the 45 days, 60 days or 90 days tenure program. So about the registration process, when you'll scroll down a bit more, you will find three different tenures. I'll just quickly change the pricing here first into USD. Okay, so when you scroll down, you can see here are three different tenures in which anyone can like register or enroll for. Now the question arises, what are the common resources which you'll have access to, and what is the difference between the tenures, the three different tenures which you all can see on the screen? So the first question, that is the common resources which you'll have access to, is online tutorials, project examples, coding sessions, session recordings, online mentor support, online technical support, certificate after completion of the program, and then you will have access to the example cloud pipelines on the Bioinfo server. So these are some of the common resources which everyone would have access to, whosoever is enrolled in the program. Now, the second question is, what is the difference between the three tenures? When we talk about the 45 days tenure, so in the 45 days tenure, you will go through with a full-fledged training related to what is bioinformatics, what are the different terminologies used in bioinformatics, what is different healthcare application where bioinformatics is being used, how bioinformatics is being used in translational research, then moving further with multi-omics studies, genomics, transcriptomics, single cell, metagenomics, microbiome, etc., and then correlating it with some machine learning, and in the end case studies and projects examples. So whatever you have learned conceptually and then practically hands-on, so you will be applying that in a real life application by converting them into an independent research project. So the 45 days is just a full-fledged training. Now, when you talk about 60 days, you will have two different opportunities. One would be one-on-one mentor sessions. So, for example, if you're going through with any of your module and you get stuck somewhere and the next live session or the live Q&A session is maybe next week or so, and you want to immediately connect with the mentor, get your queries resolved and move further with the recordings or the associated online resources or any kind of assignment. So you will have access to one-on-one mentor session in 60 days and not in 45 days. In 45 days, there are access to the group discussions and group live sessions. Then in 60 days, you will be having access to the draft, or like you can draft a research proposal presented to mentor for the evaluation purpose. So the first 45 days would be your training part, and the next 15 days would be devoted to drafting a research proposal, presenting to the mentor. Mentor will evaluate, mentor will guide you, give you a feedback, expert feedback would be there. if any amendments would be required to make it more validated. And then after that, if it's validated, you can start working on your research proposal and then convert it into a full-fledged research project. Now in 90 days, you will again have two different opportunities. One is obviously the one-on-one mentor sessions, and second is you will have an opportunity to draft your own independent research project. So the first 45 days again would be devoted to your training part, and the next 45 days would be devoted to your draftment of the independent research project. Literature review, data set finalization, proper experimental design, then analysis, interpretation, biological, statistical, and then the manuscript, etc. So this structure and or uh, this this will be done in your 90 days tenure. So once your project is completed, you will also have a chance to earn a certificate of excellence in 90 days. Now talking about the outcomes. So the first outcome is obviously the research publication when we talk about in 90 days, and then is a poster presentation competition in different research symposium and then international conference. And also we Pine Biotech, we organize biannual Omics Research Symposium, where anyone, maybe like a student, a high schooler with an example like good research project, or a graduate, a postgraduate, or like a researcher or a faculty, anyone who is interested and they have a project in their hand, they can develop a poster present, they can showcase in our research symposium. We have a proper competition and you can earn like exciting cash prizes also. So we'll be telling you about it in coming few days. So this, like, the Omics Research Symposium is in March. So we have started the registration also. We can share it afterwards through like the mails. Now this was about the 45, 60, 90 days resources you will have access to. Now coming on the pricing part. So for USD, like for those who are living in US, maybe Canada, etc. places, the pricing for 45 is $150. The 60 days it's $300, and for 90 days it's $450. And right now we are providing scholarships up to 50%. So those whosoever is interested, please ping their email address in the chat box, and our representative will mail you regarding the steps to register with a particular coupon code. So please ping your email address in the chat box. Now, if you are from India or any other country, you can change your currency accordingly. If you are from Brazil, you can change it accordingly. If you are from some European country or so, you can change it in euros. Then if, for Indian, it's in INR. And the pricing for Indian people is for 45 days, it's 7,500 INR. 60, it's 10,000 INR. And 90, it's 15,000 INR. And for Nigerian people, it's for 45 days it's naira 50,000, for 60 it's naira 1 lakh, and then for 90,000 90 days it's naira 1,50,000. So yes, again we are providing with scholarships. So those whosoever is interested, please ping your email address in the chat, and we'll connect and mail you accordingly and provide you with a better solution. So this was about the resources, the tenures, the pricing, etc. Now we have other alternative method also, like bank transfer, PayPal is there, and then UPay option for Indian community, etc. is also available. So if anyone is facing any problem related to payment process, they can mail us at marketing@omicslogic.com, or they can ping me on the official number, that is +919876 134 Hopefully, I guess Sri Gauri or maybe Shubham has already pasted the information in the chat box. Now I'll just quickly show you the procedure of the payment, how you can proceed further. For example, if someone is interested in 90 days, you need to simply click on Buy Now button, and you will be redirected to the payment page, the checkout page. If there is any coupon code, you can simply write the coupon code here, click on Apply button. The coupon code will be applied successfully. A pop-up message would showcased here on the screen. Then you need to fill out the user details, your name, your email, your phone number, your country, occupation, organization, etc. Click on the checkbox and proceed with the payment method, either PayPal or Stripe, etc. And the payment would be processed, it would be done, and you'll receive a receipt automatically if you'll pay through the portal. So this was about the payment checkout and how one can do and proceed with the payment. So we will be sharing the session recording with everyone because of the technical issues, and you can see the session recordings by tomorrow in your inbox. Secondly, those who are interested and want to continue and want to know what's there in the second session about genomic variants, DNA mutations, etc. So please hurry up and enroll yourself, and so as to leverage different resources and get to know more about bioinformatics and multi-omics field. Thank you. And if anyone is having any question related to registration, resources, scholarship, etc., please feel free to ping in the chat box. Thank you. Over to you, Sri Gauri.
Thank you so much, Sparsh. We have a query from Rosen. Does this program include practical training like how to run NGS analysis?
So yes, Rosan, this program will include practical hands-on. We have a platform. Shubham, could you please paste the platform link in the chat box? We have a platform called T-Bioinfo Server, and it's an AI-based cloud pipeline where there are different toolkits, Roseanne, and you will have particular data sets, etc., which you can like upload if you'll have excess of 60 or 90 days, and you can run those pipelines and analyze those results, interpretation, analyze those results, come up with some biological or either like statistical interpretation. If you're enrolled in 90 days, you can come up with a proper research project also. So this program includes both conceptual, theoretical plus practical part. So the Omics Logic Learn portal, which I just showed you, is for your theoretical and conceptual knowledge. And then the platform which I'm talking right now is for your practical hands-on part.
Thank you so much, Sparsh. And Rosen is also asking, can you show the price in INR?
Yeah, sure. I'll just once again share my screen and show the pricing in INR. So just a moment, it's loading. Now I guess my screen is visible. So the pricing in INR is, I'll just quickly change. So the pricing in INR for 45 days is 7,500. For 60 days, it's INR 10,000, and for 90 days, it's INR 15,000. And again, I'm telling that yes, we are providing with scholarship. So you can mail us or you can connect with me on the official number. Sri Gauri will once again paste the details here in the chat. You can save the details somewhere and you can connect with me. And I can tell you the appropriate scholarship which you'll have access to.
Well, thank you so much, Sparsh. And we do have some queries about scholarship. For that, Shubham is asking all of you to share your email IDs in the chat so that he can forward you the details. And Nashmia is asking, can you please email us the information about resources available to us?
Nashmia, I guess you are a program participant and you have enrolled in this program, if I'm not wrong. And definitely our project manager or a community manager would email you the resources available related to this program by tomorrow.
Perfect. And Rosen Roy is asking, thank you. Do I have to install any softwares for performing analysis?
No, Rosa. You don't have to install any software particularly to do the for performing any analysis. We have our own platform. You will when you'll enroll in this program, we will be providing you with some credentials, a username and a password. You need to just log in the platform and everything, like we will properly guide you how you can use, how to run a pipeline, etc. So no other softwares you need to install.
Thank you so much, Sparsh. And Shubham has shared a step-by-step manner how you can enroll for the program. And then Charles is asking, I would like to know about the feasibility of enrolling in more than one program.
So Charles, like I would suggest, if you can, like give minimum of 2 hours per day or so, 1 to 2 hours per day for each program, then definitely you can enroll more than one program. I would say not more than two, because then it would become more difficult for you to like simultaneously work on both the programs and attend sessions and complete the assignments, etc. on time. So either one or two would be better, and so that you it becomes more feasible for you to complete the sessions of both the programs and maintain the pace. So regarding, if I'm not wrong, I guess you're talking about the scholarship or so also in terms of feasibility, then yes, definitely if someone is enrolling in more than two programs, we will be providing with a good like scholarship. And also for the Nigerian community, we are already providing with the 50% scholarship. So I guess like 50% scholarship is, it's a partial scholarship and it's great enough.
And we have one more query from Nashmia. Will we be getting the recording of these lectures?
Yes, yes. Nashmia, you are a program participant, and you will have access to this program page, and you will be finding each and every session recording here on the program page itself. If I'm not wrong, you are a program participant, I guess.
And we have one final query in the chat from Nelson. Does this webinar issue a e-certificate?
Sorry, does this webinar?
Issue an e-certificate?
No, the webinar doesn't issue an e-certificate, but yes, if you'll enroll in this program, you will definitely earn an e-certificate.
All right, that was all in the queries in the chat. So if the participants have any further queries, let us know and we'll be happy to answer. Rosen is asking, I am not a participant yet. Will I get the video lecture?
So yes, you will definitely get the video lecture for this first session. As first session for every program, it's open for all, so that everyone should get a glimpse of what we'll be including in terms of module or something throughout the program. So it's free for all, it's accessible to all, and we'll be sharing the session recording with everyone. So you will also receive it.
All right, so that, I guess that was all the questions that we have. And if no one has any further queries, we'll be ending today's session. All right. Thank you everyone for joining today's session. And those of you who have not enrolled for the program yet, kindly enroll, and we hope to see you all for the next session as well. Thank you everyone. Have a great day.
Thank you, Sri Gauri. Thank you, everyone. Bye-bye. Take